All right, we're ready.
All right, good. Hello, everyone. Looks like we got a small group for now, but people will probably trickle in eventually. But I'm Daniel. I'm a PhD student at the University of California, Riverside. I've been here for 4 years. I started off trying to study pure water, and now I'm studying how insects spread a crop disease. So I'm going to show you several things today, and it's really a tribute to how diverse careers can be in STEM. Just so many ways you can go with it, and I like the freedom of that. So let me go ahead and share my screen here. All right, can you see my PowerPoint? I think so, right?
Yes.
Okay, good. So engineering meets biology, that's me. I'm an engineer studying biology, and I look at how insects spread a crop disease. So I've done a lot of different things in my career, but one of the things that guides my decisions is looking at my skills, interests, and values. So I think that's a valuable thing to talk about, and I think that's important for people to think about when they look at like jobs they might want to do in general. What, like asking yourself, what are your skills, what are your values, and what are your interests? For me, that's chemistry, physics, and math. Those are things I'm good at. Interests, I want to do something for the world, maybe, make clean water or help reduce the impacts of plant diseases. Things I value is independence, being able to decide what I do with my work, my workplace environment. I want to work in a place where there's no bullying, and I want to earn money, right? So for me, these are some of my top things that I think about when I'm deciding a career. But I want to hear from you. Could you type in the chat and let me know what are some, one or two of your skills, interests, and value, and maybe a job that might match with those. So I'm going to stop sharing for a second, and I want to hear from you in the chat.
I know you can think of something.
Yeah,
It's Friday, we know. Oh, Marcus is there. Go ahead, Marcus.
yeah, you want to hear like what field we would want to go into,
Sure.
what we like
Yeah.
about the field.
Yeah, and then how does that align with your skills, interests, and values?
Oh, okay.
Cool. Thanks for that, Marcus. Game design, usually people are super friendly in that field and creative. I would say I'm all of those. Cool. Yep. Friendly people, creative people, good things to be. Anybody else want to jump in the chat?
Wait, say something about yourself.
Yeah. Tell me about maybe some of your skills or interests or values, one or two of those, and a job that you might be able to use that for. Art and math. Cool. That's a unique combination there. Sports, baseball, good. Oh, sorry, basketball, not baseball. Misread that. Okay, cool. Math, got it. I'm gonna share my screen here again. All right, so we talked a little bit about skills, interests, and values, and thinking about jobs that might fit those. So for me, these things line up with a career in research. That's a good fit for me. I can use, I'm studying my PhD in chemical and environmental engineering. There's a lot of chemistry, physics, and math. I'm contributing to meaningful problems, and I get a lot of independence and things like that. So those things led me to UCR. That's University of California, Riverside. And just to give a plug for UCR, I'm in the chemical and environmental engineering department. We've got several research focuses that might be interesting, like computation and molecular engineering, biotechnology and biomolecular engineering, advanced materials and nanotechnology, energy conversion and storage, air quality systems engineering, and here's the one that I studied for a while, which is water quality systems engineering. Okay, so I'm going to give you just like a crude sketch of something I studied that's a bit of an advanced topic, so I want to break it down real quick. When I came to UCR, I was studying water purification, and there's this specific aspect of it that I sort of specialized in, which is colloidal particles. And you can think about it like this is a river, and this is the sediment down at the bottom of the river. If you throw a clump of dirt into the river, then it's going to settle to the bottom. But if you throw like little like microscopic objects into the river, then they might not actually go down to the bottom because they're so small that gravity is not super important. And that's where they're called colloidal particles. If they have a dimension that's less than 1,000 nanometers and they are dispersed, then we would call them colloidal particles. And their small size gives them unique properties. So like if this is a contaminant, like a bacteria, a virus, those can be colloidal particles, or just like man-made nanoparticles that are in the products that we use. Those can be nanoparticles. I mean, they can be colloidal particles. And so the thing is that these types of particles, depending on their properties, they might just go wherever the water goes. So maybe like a company accidentally pollutes some nanoparticles into a river or something like that, and then it could end up miles and miles away because just these small particles can stay suspended for a long time. And if they reach the right environments along the way, they could also concentrate in one place, right? So maybe the company didn't pollute much. They say, oh, it'll get diluted by a river, but actually they might end up getting in one spot and really affect the environment. So studying these types of chemicals can be important because they're not as intuitive for understanding their transport in the environment. And a little bit more of that sciencey background is gravity not super important because they're so small, but the charge interactions are. So here's that same particle I showed you before, and here's like a surface, maybe the sediment or the walls of the river, and oftentimes they're both going to be negatively charged on the microscopic scale. So in the water, anything that's positively charged is going to group together with those negatively charged things because opposites attract. And so like let's say there's a little bit of sodium in the river, a little bit of potassium in the river. Those things are positively charged in water, so they'll surround particle and their surfaces of the river. And then when those two things approach, that's where you get some repulsion because the positives are overlapping with the positive. And that's where that decides whether a colloidal particle is going to stay in the water or whether it's going to get attached to a surface and stick in one place. Because these overlapping charges make the two repel each other. But for those of you that know about chemistry, you might know that the van der Waals forces are going to make them attract. And if you don't know that, that's all right. But the point is that there's chemistry here, and that chemistry of the charges decides whether a particle is going to go a long distance or a short distance in the environment. So that brings me to the application of my research, of my early research. It has to do with this E. coli outbreak in 2018. I think you might have heard news about it. So let me know in the chat. Did you hear about this E. coli outbreak where they said don't eat romaine lettuce? And if so, what did you hear about it? Nathan didn't hear about it. Cool. Oliver heard about it. Mari heard about it. Nice, nice. Good. So this is like a common thing. A lot of, okay. Interesting. So it was a big deal. Actually, like over 100 people were hospitalized. Some people even died from eating this contaminated romaine lettuce. Really big deal. And so the thing was that the lettuce was contaminated with E. coli. It's a type of bacteria. It was a specific type of E. coli, and so when things like that happen, people try to study like how the bacteria even got there in the first place, right? Because if we understand how that happened, then maybe we can find a way to stop that from happening again. So I'm going to tell you about one potential way that those things happen. The dangerous bacteria often comes from like animal poop. So this is just a picture of a cow pooping, a crude picture of a cow pooping. And so if you imagine for a second that it's raining, you got animals on a farm with dangerous bacteria in their waste, then that bacteria can be picked up by this water and swept down into soil depths. And eventually it could reach a water body and it can contaminate well water or even irrigation water and then get spread on crops, right? So in my research, I studied this process here. I made a model system and then I looked at how different factors affect how far that bacteria transports in the soil with the idea of, hey, maybe if we understand it, then we can prevent it from happening. So I filled the tube with sand and I pumped bacteria contaminated water through that sand. And then I pumped some more water without bacteria in it. And what I found was there was little bacteria released. But when I added a specific type of nanoparticle to the system, which is a type of colloidal particle, really small particle, then there was bacteria release. And that was important finding because these are things that are in the environment, like we talked about before. And so this can have policy regulations, right? Like if nanoparticles being here is a problem, then policymakers might need to know, like, should I regulate whether nanoparticles can be in this place, or should I regulate the distance between livestock and well water, right? So people are going to have to study that more, but for now, the study that I did does indicate that it could be a problem. Okay, so actually what I've shown you so far is pretty traditional engineering studying, like pretty traditional engineering research, because I've been doing like talking about fluid flow, I've been talking about physics and chemistry. Those are pretty traditional engineering, which is what I did for about the first year and a half of my PhD. But then one day the professor that gives me advice about my research emails me and she says, hey, like, you can get money if you're willing to research something else. And that's a big deal for PhD students because we actually get paid to do research. Like and when you're getting your bachelor's degree, you have to pay for school unless you get scholarships or whatever. But a lot of people that do their PhD actually get paid to study. And I'm one of those people. And it's really common in my field to do that. So people pay for my research as well as paying my paycheck. It's not much, but it does pay. And because of that, that means when I do research, I have to make sure I make those people happy. So when I heard about an opportunity to get funding for my research, then I jumped on it, right? So my advisor tells me, if you do research with insects, then you can get more money. And she says, I know this sounds far from our work, but I wonder if. And this is one of those moments in STEM where like things can completely change. It's it's uh, it's such a diverse field and it's so fluid. Like people talk about their career sometimes as if they're going from point A to point B, but that has not been my experience. That's not what a lot of people have experienced. It's sometimes you go from like point A and then you go all over the place, then you get to point B. And that's what happened here. I saw funding. My advisor told me about an idea related to insects and I jumped on it. So now I study how a crop disease is spread by insects. This is a blue-green sharpshooter. It's about 1/4 of an inch long, and it's one of the major insects that spread Pierce's disease of grapevines, which cost California over $100 million per year. So you can see here that the disease shrivels up the grapes. It makes the leaves fall off. It gives some weird coloration on the stem. It's a problem. Like actually Europe's having a big problem with it right now. It affects not just grapes, but also like almonds, plums, peaches, citrus, and it could cause billions of dollars of damage in Europe over the next 50 years or so. So actually this disease is also caused by a bacteria. The bacteria survives in the mouth parts of the insects, and as the insects feed on plants, they spread the disease. So they'll feed on one plant that has the bacteria, and then they carry it with it to the next plant to then that next plant will get the bacteria. And so when the insects are feeding, they actually pierce plant tissue with their proboscis. Big word, proboscis. So it's shown by the arrow and you can see it on the next slide. So this diagram illustrates the proboscis piercing plant tissue during insect feeding. Up top, we have the insect. Down on the bottom is plants. So specifically, the insects pierce the plant tissue with their proboscis, and there's one part called the stylet that pierces through, and then it sucks out the plant juices, right? So specifically, some of you might know what xylem is, but it sucks out the xylem sap, sucks that sap up into its functional foregut, which is inside its head, and then it spits that sap out over and over again, sucks it in, spits it out, sucks it in, spits it out. And this is part of the way it feeds, but it's actually during that process that the bacteria is transmitted from the insect into the plant. Yeah. And so scientists and engineers are researching this process with the hope of finding targets for fighting the plant diseases caused by that bacteria. And the research I'm going to show you next indicates that fluid dynamics during the spitting out process likely play a major role in how that disease is transmitted. So I created a 3D model of the functional foregut, and I simulated fluid dynamics inside of that 3D model. And what I found is that the flow rates inside of the insect are super high, actually, high enough to detach bacteria, but that's not really easy to show you at the current magnification of the model, because the portion of interest is here, and the bacteria are on the inner walls, which are still not visible. So I'm going to show you what happens if you take a mold of the magenta and teal portions of the model. So basically, this is what happened if you were to like put Play-Doh or something on here and just like around the outside and get an exact representation of the magenta and teal portions just inside out. So this is the place in the insect where the disease is transmitted from. And so this is the place that I looked at more closely. What I did was I plotted the drag forces within that part of the insect. And so each of these dots represents the drag force that acts on a bacteria that is attached to the walls of the insect. So let's take a break and talk about what drag forces are. Who can tell me what they think drag forces are? Does anybody know what they are? Who's ever like you're driving down the highway, or your parents are driving down the highway, you stick your hand out the window and you feel it get pushed back by the wind? Anybody do that? I do that. So that's drag force. Anybody else have an example of what drag force might be? Drag force is when wind or water pushes back on something that's moving through it. So can anybody else think of another example of drag force?
We learned in a different STEAM meeting how scientists, I don't know if this is drag force, but they use a needle, a really small needle, to discover bumps on a certain sort of a surface so that they can see the atoms.
Oh, cool. So like atomic force microscopy. Cool. That's good. That's a force. Yeah. Like when an airplane flies through the air, it gets pushed back by the air. When it's a really windy day, you're outside and then you got to kind of lean into the wind just to walk. That's drag force. So that's what I'm talking about here. I'm saying that, oh, let me share my screen. Yep. All right. All right, so I'm saying that fluid flows through here. This is actually water flowing through here. And that I predicted the drag forces that would act on bacteria that are attached to the surface. Okay, so red is the higher drag forces, and then blue is the lower drag forces. So the good thing for me is that people have already done laboratory experiments where they watched bacteria and saw how much drag force is required to make that bacteria detached. And so that amount of drag force is shown in blue, and then the higher drag forces are more than what's required. So what you can see is that all throughout this space, almost the whole thing, the drag forces are high enough to detach bacteria. Okay, so that's one line of evidence, right? I had this hypothesis that fluid flow, I'll show you again. Yeah, I had this hypothesis that fluid flow detaches bacteria from here, and then it gets spit into the plant. So that's one line of evidence I have to support my hypothesis, that these drag forces that are predicted are high enough to detach bacteria. Let's talk about another line of evidence that I have. These diagrams here are measurements of how often bacteria are observed in different regions of this space. So the blue dots mean that bacteria are often observed at that location. Yellow means that bacteria is seldom observed, and blank means that the bacteria is never observed there. So this rectangle on the left corresponds with the simulation on the left, and then the rectangle on the right corresponds with the simulation on the right. So, and what I want you to notice is that these colonization patterns mirror these fluid dynamic simulations. So for example, in this rectangle on the left, down at the bottom, we've got this cluster of the darker blue circles, meaning that bacteria are often observed, and that corresponds with the simulation on the left where there's lots of blue. And so that makes sense that there's low predicted drag forces and a lot of bacteria remain attached, meaning that fluid flow just doesn't completely detach them. Similarly, on the rectangle on the right, we've got darker blue down here, and that's relatively low drag forces are predicted. And then in the middle, this one kind of like looks like a saddle, like on a horse where it's high in the middle, low on the sides. So this is high drag forces in the middle, and then low drag forces above and below it. And that mirrors this pattern here where we see lots of yellow in the middle, meaning not much bacteria, and then a little bit more blue above and below that. So it does kind of mirror what we see here. And then finally, up top, not a lot of bacteria up here, but we do see really high predicted drag forces. So altogether, these colonization patterns, where the bacteria attach, it mirrors the fluid dynamic predictions. And we said that the fluid dynamics are predicted to detach bacteria just based on how big the drag forces are. So all this together leads us to believe that fluid flow physically detaches bacteria. And we're going to return to that image from before. We're saying that fluid flow physically detaches bacteria from here so that it can be spit into the plant and cause disease. And so that's important because other people are said that different things are happening. So that's sort of an iterative process we have in science. People say, we think this is happening. Other people we say, we think that is happening. So we have to test it, figure out who's right, or maybe both people are right, and then hopefully find ways to solve problems with it. Yeah, so that's what we did. So before we were talking about what happens when there's bacteria inside the insect and gets spit into the plant. But before the insect ever has bacteria, it first has to feed on a plant that already has the bacteria itself, so that it can acquire the bacteria and then spread it to the next plant. So next we're going to talk about fluid dynamics inside of the plant, both during insect feeding and before the insect ever arrives. So that's going to be the next graph. And this is going to be the most complicated graph of the whole presentation. So I'm going to walk you through it. On the x-axis, we have the diameter of these xylem vessels. And on the y-axis, we have the predicted drag forces acting on bacteria. And this is a log graph, which is not necessary for understanding what I'm going to talk about next. But anything above this dark blue line is high enough to detach the bacteria, and then up here is even higher. So these triangles here represent before the insect ever arrives. We're predicting that drag forces are not high enough to detach these individual bacteria on the surface, but because of some other research, we do think it's high enough to detach the bacteria once the biofilms start to grow on the surface, like bacteria divides, forms some sticky stuff to keep the bacteria, grows taller, grows wider. And then we do think that bacteria detaches when it reaches that stage, just not when it's an individual bacteria. So now I'm going to add an extra set of data to the graph. And that is the drag forces predicted when the insect sucks in the plant juices. But these are the drag forces inside the plant that are caused by the insect feeding. So we'll notice that these are higher than before the insect arrives. When the like over in this left section of the graph, so like less than 80 micrometer diameter, and the slope is negative. One more set of data is when the insect spits into the plant. So this is the highest predicted drag forces. Again, slope is negative, and the drag forces are higher than before the insect arrives when the diameter is less than about 130, 140 micrometers. So what this shows is that during insect feeding, the fluid drag forces are high enough to detach bacteria in the smaller vessels. Okay, like the smaller they are, the more it could detach bacteria, right? And that has significance because that's how the bacteria gets acquired, gets detached from the vessel walls and then sucked up into the insect. So I'm going to sort of summarize all these results in one figure. This is a set of 3D models that are simplified, like here's the insect, here's the stylet that penetrates plant tissue, and then this is the xylem vessel, right? And then the figure A shows when the insect is spitting into the plant, the bacteria are detaching from the functional foregut, getting spit into the plant, and also getting detached from the xylem walls all at the same time. Then when the insect is sucking in, that bacteria that was already detached when the insect spit out, then it gets sucked in. And before the insect ever arrives, then the bacteria get detached probably from biofilms instead of individual cells, and they detach as these clumps that we call aggregates. So this model is important because once we have a model, then we can test the model and see like, okay, if we change one thing, then theoretically, how would that change the spread of the disease? Oh, that would be a good thing, right? If it's a good thing, then we can test it. So here's an example. This figure here summarizes all of the insect work and some of the ways that we're proposing could be ways to fight these diseases, right? So simulated fluid dynamics inside the insect, inside the plant. And so we here have a grapevine. This is a xylem vessel of a grapevine, and up here is the insect. So first way to potentially fight disease is by optimizing the diameter of the xylem vessels. So this can be accomplished like when grapevines are prepared for cultivation commercially, they're often like crossbred. So you breed one breed of grapevine with another, and then you get some sort of desirable trait. And so it could be like xylem vessel diameter, because we showed from the graphs that the smaller the xylem vessels are, then the higher the drag forces during the insect feeding. So that could mean that more bacteria are acquired by the insects. But at the same time, before the insect ever gets there, then the wider the vessel, the more the bacteria spreads within the plant because there's higher drag forces. So the disease can progress quicker. So too small is bad, too large is bad. So we're saying that there might be some sort of optimal diameter that could be better for not spreading the disease. And that's something that somebody else would have to test. Another thing is these air bubbles in the xylem vessels. So when people don't water their grapes enough, like they do in certain types of grape cultivation, that's like a practice. Like they purposely don't water their grapes enough because it creates some sort of desirable traits in the grapes. And the thing is that can make these things called embolisms, which are actually air bubbles inside the xylem vessels. And we know from physics that when an air bubble like travels across the surface of something, if a bacteria is attached, then it exerts a very large detachment force. And because we predicted our drag forces here, in the insect and the plant, we can actually say that these air bubbles would cause much higher drag forces than these drag forces and most of these drag forces. So we're saying that maybe the insect isn't even the key driver of how this disease is spread. Maybe it has to do more with these air bubbles that are caused by how little people are watering their crops. It's something that could be tested. And then another thing is that we showed it's probably when the insect spits into the plant that there's these key things for disease transmission. And so altering those behaviors could be important because like if the insect, in a hypothetical situation, if you took away its instincts and just like it's just an imaginary insect where all it has to do is drink the fluid, it doesn't spit back, then that could like reduce the transmission like crazy. So what we want to do is reduce the amount that the insect spits back into the plant because that's a key driver of the disease transmission. And it's been shown that those types of behaviors can be affected through like xylem sap chemistry and other properties. So overall, that's sort of what came out of our research. We did some fundamental engineering stuff and answered some biology questions. And we're actually pretty happy with these results. And really the reason why we were able to acquire them is because I'm an engineer. This is mainly my research, and I think very differently than the people that I work with. And that's just been like really helpful. So diversity of the workplace has been valuable. And so this is just another testament to how having people that think differently work on the same problem can be helpful. I'm going to stop sharing my screen because that's the end of my presentation. Any questions?
Yeah, I have one.
Yeah.
I have one. How did you narrow down to a specific insect?
Oh, there's a lot of them, but it's mainly based on how much do they spread the disease and how well are they studied. So there's one insect that actually spreads the disease more than this one, but it's not very well studied. There's not a lot of information published. So it's just a matter of convenience that we studied this one. If funding agencies said, okay, we think that what you did is valuable, then we would test other insects as well. But yeah, lots to learn with other insects.
Did you study the insect um, in the lab, or did you just use data for the most part?
Mostly data. We worked with people who do work in the lab, but with COVID and with things moving around with labs and stuff, the virtual stuff just worked better for us. So yeah, mostly just stuck with data. It's a big deal, guys. COVID is changing the way research is happening.
Yes, a lot of things. We got one question in the chat.
Sorry. From Isabel, thanks for the question. What is the next research project you hope to do? If I were to continue, then I would work on a project applying this to other insects, really, and making some observations in plants. So really, it was a question of me saying, like, who's willing to fund the research and what do they want, right? So the California Department of Food and Agriculture says that they want to know research on this other specific insect, and so I decided. So I wrote a proposal on that insect, and I spinned a story that I thought they would want, and got some funding that I probably won't. I'm not sure if I'm going to follow through on it, but yeah, it's all based on what does the funding agency have to offer. I would do some experiments where I looked at plant properties and insect properties and see if my model was actually correct in predicting how those things happened. Thanks for the active chat, everyone. So drag forces like resistance. Yep. Planes flying. Good one.
Oh,
Sorry.
that's okay. I didn't see the chat. Did you answer that question? I have one. What region is that insect primarily from?
Blue-green sharpshooter is a big deal in California. Back before early 2000s, that was the biggest deal. And then there was an invasive species called the glassy wing sharpshooter that changed the game completely. So this newer insect, I mean, relatively new, it's been here for decades, but glassy wing sharpshooter can fly a lot farther. It can feed on the woody portions of plants, whereas the one that I studied only feeds on the green portions, so it's not as strong. And so, because it can fly farther, the disease spreads faster, and the way that grapevines are grown, that every year they cut back all the green stuff so that it ends up just the woody stump, and then the next year the green stuff grows back in these commercial vineyards. So the glassy wing sharpshooter can still keep on feeding and spreading the disease even when they cut everything back. So it was a huge deal. Yep.
So is that the one around getting in the forest?
Sorry.
Or is that the one that's getting the trees in the forest, or is that some beetle or something?
All trees in the forest. I would imagine that's probably a different one, but is there some like that happening right now?
I think it's a beetle that's going around killing all of the forest trees.
Sounds like
heard. Yeah, I think Sophia and Isabel had a question, so let me get back to them.
Yes, I had two. One of them was that, like, is there specific or multiple specific plants that a certain insect like targets?
Yeah, it has a feeding preference actually based on nitrogen content. It's funny because like humans, like we're kind of obsessed with protein more than our nutritional requirements. Like people are like, oh, I need protein. I'm gonna work out. I'm gonna eat protein. These insects are kind of obsessed with the protein. They see amino acids in the plant, and then they go for it. Like the more amino acids is better. And then so that's where it does attract to specific plants. But the thing is, the bacteria itself is only pathogenic to some plants. So the bacteria has like a specific lifestyle where it like multiplies a certain amount, it spreads a certain amount, and it's only a problem for some plants. So it's a combination of the insect feeding preference and the bacteria behavior that decides whether it's going to be a problem or not.
Thank you. And what is your favorite part about your job?
Favorite part? 3D modeling, easy. Anybody here play with Legos at some point in their life? Good. Yeah, I did too. All right. I love Legos. And one of the things I really like about Legos is that I can have something in my mind I want to build. And I can use like rectangles and cylinders and squares, put them together and make a whole different shape, like a spaceship or something. And I got to return to that like childish enjoyment in my work for a couple of months as my whole job was just to make a 3D model inside this insect. And it's a skill that not a lot of people have. So yeah, if you like Legos, you might like 3D modeling, and within the right fields, it could be helpful for the work. Thanks for the questions, guys. Good questions.
So what kind of careers can you get in this field?
All sorts. So the broad fields would be like industry, government, or academia, right? If like most people that go for a PhD have some sort of academia in mind at the start, but the reality is that by the time they graduate, most people don't go into academia because like there's only a certain amount of professor positions, and in general, like there are generally like the same amount of professor positions every year. So like if you have a professor and they train, they have one graduate student, one PhD student graduate every year, and over their whole career they might have 15 PhD students, then statistically speaking, then only one of those 15 PhD students can then become a professor, right? Because there's only just so many jobs out there. So with this work has to do with crop disease, you could go into the government, work with the USDA, and help like set regulations for fighting the disease and help inform farmers best practices. You might go into the Environmental Protection Agency, and they form regulations on the spread of contaminants in the environment. And then industry, I'm trying to think. I don't super know a lot about industry, but when I'm applying for industry jobs these days, it's mostly just based on the skills I have rather than the subject of the research. Yep. But lots of people, I mean, it's an important problem, right? It's a problem that's not going to go away. There's always going to be diseases in crops, just like there's always going to be people diseases. So we're always going to need doctors, and we're always going to need people that fight crop diseases.
What kind of courses should the kids take?
For me, I'm a chemical engineer, so the foundation of the courses would be chemistry, physics, and math. But the name of my courses are like specific areas within those. So it would be kinetics, thermodynamics, transport, and computation. So we do a lot of computer programming. We do a lot of calculus. We do a lot of analyzing things from a systems perspective, where we say there's this much input and that much output. So, what changed? That's really a lot what of our course is about. It's a tough thing, but a lot of good jobs.
Any of you kids interested in chemical engineering? I know there was it appears to be a student from UCR on here also. So.
Oh yeah.
Yeah. Any more questions?
Chemical engineering in general is one of the harder among engineering. And usually people go into this field because they like money. Because chemistry, physics, and math are three things that a lot of people don't like to do, or they're not good at it. And so if you can tolerate it, if it's somewhat pleasant to do it, and your career is about money, then that's typically why people go into it, even though they might not admit it. They might say it's their passion, but when it gets down to it, I think that's really why they're here. It's a lucrative career.
Yeah, sorry, I came in a little bit late, so I don't know if you already talked about this, but like, have you done any other projects, and if so, what have you done?
Tons of projects. As a PhD student, my goal would be, one of my goals is to get my name on as many publications as possible. So to get my name on the author list. And so I've been contacted by people at Berkeley about like doing some modeling for their projects. So I did a little bit of that. And I worked with some simulations of software. Like there's a physics simulation software that doesn't do everything I wanted it to do. So I did some computer programming to make it do what I wanted to do. Yeah, those are the ones I'm thinking of off the top of my head, but it's been piecewise. Like people tell me they want me to do work, and then I say, okay, you're gonna put my name on an authorship list. That works for me.
Now tell the students the importance of that,
Of which part?
getting your name on authoring projects or journals or some kind of manuscript.
Yeah. Yeah, I mean, getting the 3 letters PhD behind your name, that's what you essentially get when you get a PhD degree. Most people don't go and do the work exactly that they study, but they get prestige, right? So when someone says, you know, Dr. So-and-so, they automatically have like esteem, like, oh, maybe I should listen to what this person says because they're probably an expert or something. And so the publications are another way of doing that. For people that don't just look at the name, they might look you up on the internet on Google Scholar and they might say, what is this person published in? Are they qualified to talk about this? Or when you're applying for jobs, the more publications you have, the better it looks. If people try to become professors, there's a strong correlation between like how much they publish and if they actually get a job or not. So basically you have to be the best of the best, publish a lot and do good research if you want to go into being a professor or something like that. In industry, the publications are not as important, but it always looks good if you have more publications. Not yet, it's coming. Great question. Oliver was clearly listening to what I was saying. No, yeah, it's coming. Yeah, probably a year from now, this stuff should be published. This stuff that I talked about. Francisco says, no, I'm into architectural engineering. Cool. Monica Cruz, I'm the engineering career specialist at UCR's Career Center. Cool, Monica. Wait, I was late. I'm in chemical and environmental engineering. Good, great, thank you. Thank you, everyone. Great, Oliver, thank you.
Wow, any more questions? Very interesting topic. I was telling my sister who's online, I said, oh my gosh, I'm going to be seeing bugs and things where where they make a bunch of holes and stuff. Because I have an issue where if I see a lot of like holes and stuff at once, I like get freaked out for some reason. So I was all like, I gotta have one eye open. but it was great. It was it was a great explanation, and people like us who really don't even understand the technology and the science behind certain things. You made it really easy to understand, and I'm fairly certain the kids would follow along, because some of them know more than I do. So,
Appreciate it.
oh, somebody wants to know about the math requirements.
I'm not too familiar with math requirements for architectural engineering. I went to a community college first, and by the time I transferred, I was supposed to have 3 classes of calculus and 1 class of differential equations. Then I was ready to take the engineering classes completely. Lots and lots of math.
There's a lot of physics involved and stuff like that. So it's, yeah, like you said, it's all the stuff people don't like to do, right?
Yep. So if you can tolerate it, then yeah, that's a good sign. If people don't like it and you think it's all right, then that means you got a potential career if you're interested.
Yes. Data science. Yes, there's a lot of math and data science.
Cool. 3 classes of Calc 2. Cool.
All right. You have anything else? Can they, if they have any questions after, is there a way we can contact? I know I can contact you, but can they contact me and I contact you for
Sure, I can
questions?
put it my email in the chat.
Okay. There you go, guys. If you have a question for Daniel later on after the video, there you go.
Yeah, like questions about like if you're ever interested in graduate school or undergrad, how do I navigate the process, good study habits, things like that. Want somebody to bounce ideas off of.
And that's great because, you know, folks my age, we didn't have that growing up. It was like, on the job training basically, or in school, but, we have all these resources now and mentors that the kids can use. So if you need help or, because some of you are in high school, some of you guys got a couple of years to go still, but, we, that's what he's here for. So he's extended his help to you guys. So by all means, utilize anybody that's willing to give you a helping hand, right?
Yeah. If people point it out, then they're probably real interested in helping.
Yes. Okay. Well, we're going to give everybody 5 minutes back. And